All right, thank you guys so much. I appreciate all of the energy, right? It's always fun to go out and experience what we have available to us and maybe even see things in a whole other way. I'm going to explain how to prepare a slide, at which point you guys will prepare the slides. My objective is not to... I mean, you guys can call me whenever you want, and we'll come and hang out on the farm. That's not a problem. But my objective is not to create a lack of flow with the information. It's to actually help you guys understand that you are totally capable 100 different ways. And most people think they have to send off for a soil sample, okay? When you send off for a soil sample, depends on what the sample is, right? The type of samples that you're taking. But when you send it off, they take whatever you sent them, right? I have bags of soil samples actually from a place just the other day. They put it into an oven and kill everything so that they could get just the minerals to tell you what you have in minerals. But what did they do in the process? It doesn't tell you anything about the biology. There's another kind of test. It's called a Haney test, Haney. The Haney test simulates nature in how you'll have a rain, you'll have a bloom, you'll have an expression of carbon dioxide. Remember, it's breathing oxygen and respirating carbon dioxide. When that happens, you get so much more information because it's directly connected to the biology at that point. Haney. His name's Rick Haney. Okay? He's a good guy. He just retired. There's lots of labs that do it. I'm not promoting one over the other. If you want to call me, I can come out to your farm and I'll send it off to them to do the test too, right? You can do that on your own. The Haney test gives you information connected to the biology, but at that point, the mineral test tells you what structure, nutrition, the soil has in it. So those two tests are paralleled. This is what I have to work with. These are the puzzle pieces. These are the creatures that want to work with it. For a couple $100, you can actually have a genomics test, and it'll tell you the genes of what's in your soil. And then if you have enough time, you can actually go back and find white papers that says, this creature right here deals with alkaline soil. It likes it. At that point, it's breaking down, metabolizing what you don't want to deal with. And at that point, that's another teammate on your behalf, right? It really is an amazing process. The website, this is my website right here, wegrowwith.co. I sell kits. These are kits. All the puzzle pieces you need so that you can maximize you as an observer. Okay? There's 3 different sections because different people use it just for themselves as a steward. That one has a Wi-Fi camera so you can look at it through the screen of your phone, take a picture of it, and you can email it to someone and say, what is this? Right? Versus, hey, I'm seeing weird stuff in here, not sure what's happening, and nobody else can see what you see. As the educator, it's a different type of camera so we can all see the same thing. And then there's a laboratory kit, which also allows you to actually look into the root system itself to determine if the fungal communities have already established a relationship. And if it has established a relationship, you don't need to buy more, right? They're already inoculated. Some of the terminology is changing in the industry right now. They're getting away from what's called the soil food web, nutrient cycling, and they're leaning more towards what's called the fungal energy channel. Have you guys heard of that at all? It's recognizing that all of this is interconnected, and the fungi are the pathway that channels all of that energy through the entire soil profile into and through your plants. It's directly connected. Sky above, processed through plant, interacted with the soil community, and it's the fungal community, the beneficial fungi that actually brings it back and through and allows it to grow. Have anybody here heard the term mycorrhizae? Okay, that's part of that fungal energy channel, right? It really is your roots being 10 times larger than the actual structure of the plant because the root is covered in even smaller little roots or filaments called hyphae that are 10 times smaller than a root hair. That's how, when it has this relationship, it can actually access the water and nutrition that the plant couldn't get through, that fungal hyphae will work through in a three-dimensional world and have access to. A saprophyte is usually understood to break down dead living material. Saprophytes, when the plant is alive, establish the epigenetics. Okay? That is actually engaging all of those systems, and then when the plant dies for whatever reason, system-wise, at that point, the plant has that biology already inside of it, which then breaks it down in nutrient cycling. Any other questions? There was a lot of questions that actually happened over break.
This question just pertains to soil samples. As we're about to see from your experiment we're going to do now, it's going to be very diverse from one piece of land. So if we have, say, an acre of farm,
yep.
a soil sample, and we're not doing it ourselves, we want mineral content.
Okay.
Are we, some say, take it from here, 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 and mix it, and then give it to them. That seems ridiculous to me.
Actually, most of the properties in the United States already have had soil samples done on them. The USGA years ago went out to the Geological Society and went through everybody's properties and looked around and said, oh, here's a sandy loam with this type of mineral profile. Here's this with this profile, right?
So I'm not going to have a mineral spectrum on 5 acres. I'm going to have a set mineral.
It's all, okay, so this is honestly where, like, me as an ambassador, it really is context. I don't know. Like, from one side of your property, maybe a ridge on this side and something else over here with a water feature that comes through and spreads out minerals from far away, right? The entire Pacific coast is a depositional beach of all the mineral nutrition from the entire Pacific Northwest coming down across, and then ends up as sand here in California and northern and in Oregon. So it really is context dependent. And so at that point, if you have two specific areas, pull from them. At that point, you'll be able to compare that, and you'll say, well, over here has a lot more iron. This area is deficient. Your job is to now take some of what's over here, put it over there, right? Instead of purchasing a product, you're purchasing the knowledge. You're paying for that perspective. Okay. With any type of test, right, it's all subject to the observer. I've had people tell me, here's an example. They'll send you, like, can I send you a soil sample? It will be a box, 40-pound box of dirt. I'm not kidding. It costs a lot of money, for reals. When they do a sample, who has one? I'm telling you guys secrets right now, so prepare yourself. This is a 15 milliliter. You fill up this base until it becomes the cylinder. This is 1 milliliter of material. I'm going to break it up a little bit because of what it is. Why would we want to use water? Puts it into suspension. Suspends it, right? Question now, what kind of water do I want to use? Why distilled? Because there's nothing else in it. So 1 milliliter of the material with 5 milliliters of water gives us a 5-to-1 ratio. Okay? I'm not kidding. This is high-end science right now, everybody. Are you preparing yourself? Do this for 30 seconds. I can give you guys a dozen labs that this is exactly what somebody's doing, and they're going to charge you hundreds of dollars. This is how you get scientific consistency, right? 1 to 5, rock it like a baby. Because if I do this, right, inconsistency, we might just obliterated everything, right? What we're trying to do is get the material that was in out, floating around, right? And then we'll take a sample of that. The other aspect of consistency, how deep do I go? I don't care. Just do it the same. So I hold it, I put it in, I blow out a bubble, bloop. I take in a drop. Guess what happens at that point? I literally just put it on a piece of glass, put another piece of glass over the top of it, and then I take a look. If you know how to count, you can quantify. That's all it is, putting numbers to it. If you don't like counting, don't do it. But still take a look. It's that simple. This is just an observation tool, just like the pair of binoculars. People are not intimidated by a pair of binoculars. Maybe, I am assuming. Anybody here intimidated by a pair of binoculars? This is a pair of binoculars connected to an Etch-a-Sketch. Okay. Who told you to take my chair? Okay. Oh, don't worry. You're all going to get a chance to sit in the hot seat. Don't worry here. All right. So the way this...
First one to go. No, just kidding.
I'm just demonstrating. Then I'm going to stand over there and answer questions. Everybody else is going to take a look. Okay? This is part of it. At this point, if you have the right tools, you can take a picture, write down information. You can say, hey, this is from corn soybean field plot 78 with this, we just added such and such to it. We're noticing a yellowing. If you're noticing a yellowing, that's because 3 days before you killed everything. Nature takes a little bit of time to catch up before it starts asking us for help. Right? Most of us, our memory isn't very good, and so we blame it on ourselves or something in the future versus realizing that it was 3 days ago we killed everything, and the plant's finally getting around to not having its team and going, hey, anybody else up there? Can they help me? Right? So, this is a slide. Just support it. Goes down. Consistent spot, though. Put one drop right in the center. Put the lid back on. That'll get you. All right? At that point, we have a slide cover or a cover slip. If you know what color your shirt is, it won't get confusing later when you go, what is that black thing? What is that? I've never seen one of those before. And you're like, oh, wait, hold on. That's my shirt. Right? Cover slide. You put it at an angle on the edge of the dot, and then you let go. Putting it on an angle at the edge of the dot, pulls the water to it, goes across the whole slide cover, you lay it down, it spreads it out. At that point, you take a look. We are fortunate enough to be able to look together. Okay? We might actually get to see some really cool stuff. Moss is a whole little micro-forest system with creatures called tardigrades in them. I don't know if you guys ever... They have 8 legs. They're super cool. They're supposed to be, like, indestructible. They've taken them to space. They're called water bears. Okay. You can see all of this with your naked eye or a pair of a magnifying glass, right? The biggest stuff is at this level. Even though they're called micros, at this scale, they're the bigger ones. You have different roles within the ecosystem. You have microarthropods. Arthropods are just like little type insects, right? They're shredders. Definitely would find them in the leaf mulch. They're shredding up that material. That's why it has that fluffy consistency. They're breaking it up. Bacteria we will not see until we hit 400 magnification. Right now we're only at 40. Okay, so I'm going to close this because if I move it around real fast, you guys all get motion sickness and it's not good. Or we can watch and see this is how, looking over. Hey, look, there's a thread. See? Okay. So we're going to now, you literally just go side to side, down, same field of view, over the other side, down, field of view, over this other side, and you go through all the passes and you can see everything on the entire slide. It's representing 1 drop of water from 1 milliliter of soil, which potentially was sent in a 40-pound box of material, trying to get an idea of what you have to work with. The only reason why this actually works is because when you have things in water, it all balances out. It becomes a spectrum, a perspective of the whole, right? That's why when you're out in the ocean, it has just about everything in it, but it's just really little and all spread out, okay? So, 1 second. If you have really weird insect larvae, you'll see them at this phase. Right. So we're still at 40 magnification. It's a 40 because the eyepieces are 10. This is a 4x lens. We're going to go from 40 to 100. Here's something really cool, everybody. Anybody tell me what that is right there?
The lower green thing?
Yeah, green. You got the color. See, some people are colorblind, and it really gets awkward. But if it's... Why are you laughing? You guys just earned your green thumb. Congratulations. If it's green, it's photosynthesizing. That's it. If it's green, it's photosynthesizing. At that point, it's taking in sunshine and turning it into sugars. This is a little algae or part of the moss, right? I can't tell you exactly what it is. I can tell it's a plant cell because of the square structure to it, but really it's green. It's growing, right? This is also something you don't see in most agricultural fields at all. And frankly, there's more life in this picture. There's even a spore that's happening up here that doesn't exist at all. You just found it outside. It's because we're in a healthy ecosystem. The rule of I, or the role of I, is for you in your life to go to healthy ecosystems and bring it home, right? I had someone ask me in the last class I taught, how do I know? And it's because that's where I take my children. It's the healthiest beach in California, healthiest mountains in California, healthiest streams in California. Where do you guys take your kids? Your hands are on your face, right? But the perspective is that this is actually a combination of from the macro to the micro about health and healing and nurturing and cultivation of good. We're growing the good. Okay? We're going to go from 100 to 400. We're now going to start to see bacteria, right?
Before you change that, I'm not colorblind, but that does not look green to me.
Well, why don't you come up here and look?
I can see it now. Oh, thank you.
You're welcome.
I didn't think you were lying. I just thought you were lying.
You're from California, huh? It happens. So, all right. We're good? It's growing. Okay. We're going to go to 400. When we hit 400, we're actually going to see the foundation point of the nutrient cycle because we're seeing bacteria. The bacteria are mostly nitrogen. filled, like the fuel, right? If there's nothing to eat and tear open those little fertility packets of fuel, then the plant itself can peel back this outer protective layer of them and actually, through a process called rhizophagy, eat that microbe, have it go up and through its whole system, and then put it back out into the soil to keep going. It's called root feeding, rhizophagy. That's just what that means. We're going to go to 400. Oh, look at this. Hey, can you see that?
Yes, I can.
Good. Wouldn't it be awkward if it wasn't? Yeah, it would. That's a bummer. All right. At this scale, right, it's the role of I again. Sometimes when I read Scripture, I a different perspective. And so when Christ's like every dot and every, I think he meant wiggle, and it's just a typo, right? Like, can you imagine, like, every dot, those are called cocci, they're single-celled bacteria, right? Every dot and every wiggle. Be fruitful and multiply, right? These here, we're at such a scale. This is, remember, just a drop. Dark amber-colored crystals, those are called humic acid. And to be just totally honest, it's not even an acid. This is how confusing the industry is. If they take acid and pour it into good, healthy soil and they flush it through, you get fulvic acid. It's a different pH scale. If you take water, which is what we did, and do exactly what we did, it's water soluble. It's humic. Yeah, there's some really interesting little twists to this because the word Adam, right, and then the Hebrew word Adamah, it means that's made from the healthy soil, like good ground. That Adamah is humin, Humin. It's the most stable. It's the most nutritious. It's the best for the planet. We're the human, M-A-N. This is human. It's a speech impediment thing for some people, but it is really the same thing and different, okay? When we look at the hue, the color component, this is where we're talking about diversity. We're talking about the whole palette of creation, right? The human is the manager. And manage just means the old man, the man who's aged. It's true. It's kind of true. Right? So, at this point, can you guys see them? That's your labor force. They're little. Cut them some slack. Right? That labor force is waiting for something to come and eat it. They don't take it personal, like honest. At that point, whatever's in it becomes plant-available nutrition, water-soluble. That's water-soluble nutrition from a biological scale. Well, hi.
Did you see this?
You know, I am, actually. Thank you. Thank you for clarifying. At this point, I don't care who goes first or who goes next. Take a few minutes. Everybody has an opportunity to utilize this tool. The fine focus knob, larger focus, etch a sketch. Up, down, side, side. Make sense? Who's next? There you go. Look, you're ready to go. All right. All right. We're going to have a seat. We're going to go back to the 4, which creates space. We open the side, pull out the slide. Okay. Right? At this point...
Do I use the same slide? Or?
Nope, because it's dirty. Well, it's not dirty. It's been soiled. This is really like how word changes, right? Dirt's dead. I want soiled, spoiled children.
Any one of these?
Yep, you take this.
Same one?
Yeah, we're going to do it. We'll flush it. Right? Okay, there you go. If at a 1 to 5 dilution ratio, here, we'll pull out a slide.
I got one.
Oh, perfect, and then the cover slip. If at a 1 to 5 dilution ratio, you can't see anything, there's no space in between, at that point, you have to go to a higher dilution ratio, a 1 to 10, a 1 to 20, 1 to 15. If you have clay soil, where it just holds suspended, and then when it dries, it becomes concrete, at that point, you're going to have a higher dilution ratio. Those minerals are so small, they're just layered upon layer, and that's why clay doesn't allow that water. It's hydrophobic at that point. Questions while we figure the next slide out? Yes, in the back. Well, it is soil. It's alive, but it's in this form, right? So at that point, it's really the fungal component that builds and brines and breaks that up, right? They're able to work between those fine particles, and then at that point, they're able to have access and utilize it. For the people here in California, we have wildland fires. It terrifies a lot of people. If you're from other states, you might not understand how literally just some trees, on a hillside will, cause a whole community to panic. If you're out in nature and the fungal populations are healthy, you go out and step out on a log, that log, right, and turns into soil. It's just looking for us to get it closer. That's because a spore flew through the air, landed on that log, or it was already part of the epigenetics of that plant, and has eaten the hard part, the lignin and cellulose. Okay? At that point, if we have a fuel load, food, it has to be consumed by fire or by metabolism. If it's being consumed by fire, we killed the things that ate it, the metabolism. And it makes perfect sense why the entire Central Valley is pumping out hundreds, if not millions of gallons of things that kill fungi. And then it's called drift onto the entire Sierra Nevadas. And then that population is killed off, and that fuel load over decades builds up, and then it burns. If the Sierra Nevadas don't have a healthy population, then that population doesn't spread across the deserts because it's dead, which means the deserts are deserted. Make sense? Okay, this is a little bit of wordplay, but it's a desertion. Like there's nothing there. There's a system called an arid environment. It means they like living in Arizona. They like the dry heat. They can be a healthy ecosystem and habitat. Great. It's not a desert. Deserts are man-made. Arid environments are places where nature just doesn't like as much water. Right? What was that? It's you guys religious, right? Why is it called dirt devil? You guys know what a dirt devil is? What do they call it in West Africa? When you have the finest material get picked up by the wind and blown away. Do you guys have a special name for that? No? I call it a dirt devil. Dust devils, dirt devils, okay? Tornadoes, right? All right, from a nature perspective, it's the Holy Spirit coming through and saying, wow, this place sucks to live. How about I take the littlest ones with me? Right?
I wanted to show them an image of dirt devils.
No, it's kind of a funny, but the whole devil component to it really is we've sinned. We've missed the mark. And the land is leaving the life that likes to live there by land, sea, or air. That's called erosion. And when it's not bound together by the fungal component, interlocking in this network, right, it will leave. That same air patterns move through everywhere else. But it's bound together. It's held onto. At that point, that same invisible vortex passing through doesn't have anything to grab onto. And it moves on through. When it goes through ag land, it creates the dirt devil, right? It's a tattletale on your neighbor or on yourself, right? It's also indicating that you have a lack of fungal component and trace elements because they're leaving. That's the lightest material, right? And they're literally picked up, blown through weather patterns, and hoping to find a better place to live. It's habitat hospitality.
I don't know if you already covered this or not, but how do you build up the life in the soil?
That's crazy life. First of all, they make babies. Some of them don't even make babies. It literally is like, hey, let's just have two, right? At that point, you have to have the conductor that's out there 24/7, and those are the plants, right? If it looks like a crystal or mineral, it is, right? When you look at sandy soils, it's just mostly little crystals. It's beautiful, right? It looks like a little kaleidoscope. Can you go to the, yeah. So this right here.
Oops, sorry.
That's right. Okay. This right here on one of the labels that you might read would be called a propagule or a colony-forming unit. It may die, but they get to put it on a label anyways. Okay? It's a part of a fungal strand or hypha. At this scale, form is function, right? If the fungal hyphae are wide, amber in color, with what are called septa divisions between, it's beneficial, right? The water-borne fungi, or what most people deal with, like powdery mildew, things like that, doesn't look like that at all. Literally, it's thin, it's thready, it's not, structure like this. And at that point, you end up with something that is a opportunist. It's still there because just like with the hand sanitizers, we killed 99% of the good guys. The ones that are left over, waterborne pathogens, a lot of them. This is just an observation tool if you want to know what's going on in your water that you're irrigating with. If you're wanting to know what's happening upstream, this will tell you. And it also allows you the opportunity. It allows you the opportunity to compare and contrast throughout the season. These kits that,
The little guys, littlest ones.
okay, so remember that right now. So first of all, this is at 1,000 magnification. Okay. You guys see it almost looks like a little bit of a rainbow effect? It's because we're looking through them and there's a prisming of the light through the body. Okay. The smallest dots are called cocci. It pretty much just means dot, right? Bacilli means bar-shaped, right? Form and function. Yeah. If at that point you want more specificity, more details, it becomes how do they grow together? And that's why you have all the different growth plates that they use, right? But I'll tell you another secret. It's not even a secret. They tell all this stuff out there. About 99% of the life that is in the soil doesn't want to grow on a sugar plate. Does not want to be cultured in a lab. Therefore, most of the products, if it's not nature-made in vivo versus in vitro, if it's not nature-made, it doesn't want to grow there. In vivo is what people can produce at scale, which is mostly why they're fermenters, because they're shelf-stable. Most tuning forks, right? Cultivator forks have short handles with a D. Do you guys know why that is? Little handle?
Yeah.
Why? Well, at least you recognize, right? Right? At that point, the little handle is so you can fit more on a shelf at a hardware store. Who the heck wants to do this? That's not how it works, right? They're harder to find, but if you have a short-handled one, you take off that handle, put a long handle on it, it's still good to go. Why do they have the lacquer on the outside? What? No. So it looks pretty. It looks pretty and it preserves the luster while it sits on a shelf for years. I know. Most people spending money at the hardware, hard is store, think it's practical. Strip off that lacquer because the lacquer is what grinds and bites your hands. That's what gives you your blisters. You get rid of that. You put a little bit of oil into it. It breathes. It's wood. It works for you, right? A little patina to it. Works out fine. You can work with this all day long. It's a shift of perspective because you're the one getting to use it versus the person selling it to you with their own agenda and marketing and labels and pretty pictures and all the things.
How about wax candles?
Wax candles are great. Yeah. Boiled linseed, or I prefer raw linseed oil. When they boil it down, they add other chemicals to it. But it's just an oil. You can use any oil. I've lived in the tropics. Coconut oil is used for just about everything, right? Sorry. When we have here, and I'll adjust where it is real fast. I'm going to go to the other side. Good thing there's a chair there. Who would like to set up the next slide?
So that 1,000 power doesn't require oil.
It doesn't. So this company that I am a distributor for, they were able to invent a 100x, which allows 1,000 magnification dry lens. That's why I work with them. They literally were able to make it click, click, click just like normal versus you have to mess with oil, then that oil gets on things, it gets on your other lenses, it ruins everything, nobody wants to use it. Does anybody here have a microscope? So you guys share one. No, 1, 2, 3, 4, 5. Okay. How could you come to an advanced class unprepared? This is the tool that you're requiring, right? So, anybody here have a pitchfork? All right, good. At least we're right. If you don't have a pitchfork, if you don't have a microscope, the pitchfork and all your other senses will do it. But you have to fine-tune it. That's how the nose knows to say no. Because it stinks, right? Beware of the rot. It's a different ecosystem. It's the anaerobes that are producing alcohols. When you have an anaerobic environment, those alcohols will burn the root tips right off your roots. And then you wonder why your plant's dying or not thriving, right? It's still alive but not really doing anything. That alcohol literally burned them all off. And at that point, it's just trying to survive. Question? That is why the thermometer is so important. Anything over 155, 160 shifts the whole ecosystem, and it kills off what are known as mesophilic or thermophilic. There's a whole other phase to the point where I'll tell you. This is why some compost can catch on fire, because it's now filled with alcohol, and it's 175 degrees. The anaerobes produce it. It's the result of their metabolism versus aerobic metabolism. So right here, this right here is what's known as a microaggregate. Yeah. These are your microbes, right? Agora, or to come together, group, right? It requires these before we can see them on the macro scale. That's what the air and water passage is. So this tells me that there's already enough community here to start pulling together so that this... Everybody okay? Got a little... no, this is reality TV. I could hook this into the TV. This is what my kids Dad, did you see that? Right? This is that's the slide drying. Like everything shifting all at one time is the slide drying and shifting over. So don't panic.
They ran away from us.
Yeah, see? Exactly. Hopefully not. It is alive. Actually, it is alive. So just like when we were talking about, you can put in, just like we were talking about with a shift away from the soil food web to talk about the fungal energy channel, it's the understanding that the carbon itself isn't just like a chunk of wood chip in the soil. That's not how we need to sequester carbon, just like bury wood chips, right? It's called, let's see. What we're wanting to promote, which is what that aggregate is mineral associated organic matter. Okay? And depends on how you say it. You could be like, Mom, right? But this is your actual dirt that's paired with the biology and why it matters. This is sequestered carbon. It's literally the liquid carbon, the label carbon that's in that system, having the resources, and then pulling together these aggregates. That's the carbon that you're wanting. It's like a little apartment complex filled with absolutely everything. That's why they're all hanging out with each other. Those aggregates will get bigger and bigger and bigger from the microbe scale, literally single dots, right? All the way into something you can hold in your hand. Kind of looks like cottage cheese. And it's a weird soil to work with because we're not used to it. We're used to hard, right? And you can take your hand and literally down into the soil up to your elbow in good, healthy, organic matter. And then you don't even feel dirty because you're not. You've been soiled, right? It's true. That's how it works. Questions? More questions?
Yes.
All right, let's keep hearing them.
So as far as tillage goes, there was a brief mention of that at the beginning. I'm curious.
Well, why don't you tell me? What do you think tillage does?
Well, there's a lot of conflicting views, right?
Okay.
Some say the oxidation destroys all your microbial life. Some say the tillage aerates the soil to encourage the aerobic microbial life.
Okay.
And some say it depends on what your method of tillage is. Rototillers destroy everything, whereas broad forks are helpful. But, you can't broad fork 5 acres, so.
You can.
I mean, you could, but I don't want to.
Yeah, for reals. So over the last, what I'm sharing with you is part of my own personal journey and experience, both in regeneration and recovery. I lost 100 pounds. Some people talking about my shoes. Like, I realized we're springs, right? They're pumps, right? I got rid of my weed eater. Just tore me up. 4 major back surgeries, not good at all, right? Plus the cost, plus of this, plus of that. And I bought a scythe. Cost $200, weighs less than 4 pounds, and I can strengthen the core, right? And I'm done. I can come back again tomorrow. I'm done. Come back again tomorrow. When I went out there this morning and collected the dew, everything that's been cut like that is still alive. Everything that goes through a weed eater or a mower isn't at all, right? At that point, if I'm wanting to take what's called an extraction, that material, I literally can put it through an appropriate bag, right? Strainer bag. Rinse off that material. It's some of it's going to be green, but majority isn't. If you put it through a weed eater, it's just cellulose and plant juices everywhere, right? At that point, you're taking the biology that is on that material and you can put it back out into that system. With tillage, it depends on what you're working with. You have to be strategic. You're setting the optimal path. Sometimes that's flipping the hammer over and using the claw end to pull out the nail. If you have a compaction zone, that's a limiting factor. If you get rid of that limiting factor and then reset the system for that optimal path, at that point, you're not going to have the issue because you've reestablished that whole system, put all the fuses back into, and it becomes functional again. If you just obliterate it, right, and then go through the system, water, kill more things, do more, management techniques on it, at that point, you're just resetting that system for the problem again next year, the problem again next year, the problem again next year. I work with an indoor grower in Nevada. He's on his 27th cycle of indoor grown, it's cannabis, but indoor grown cannabis that is 100% just in beds. He doesn't add any fertility to it. It's alive. And he literally can take a plant with a pot with no bottom on it, put it on there, those roots spread down into it. He harvests, he goes through another pot, harvests, next to. He's done this for over 6 years, has not added one input. He makes a compost pile every year. He just adds the life, adds the diversity to it. It's a closed loop system. It's pretty amazing. Yes, sir?
Now, when you do cover cropping, how
Okay.
does that relate to tillage? How do you...
Okay, so what cover cropping is it adds a protective layer, right? You don't want the ground to be exposed. It adds diversity because each one of those plants is putting out its own little flavor into that ecosystem. And if you are going to do cover cropping, it's not necessarily about the number of types of seeds, it's the number of families of plants represented. They're showing now that if you have 4 different families of plants, that those 4 families represent such a large spectrum that at that point the diversity for the biological component is covered, versus saying we have 10 different types of clover.
Right.
You really only have one plant, right?
You're speaking of legumes, grains, etc.
Legumes, grains, grasses, asteraceae, like actual plants, like sunflowers, like the diversity at that point, but from 4 distinctive family groups. So it's really amazing because it provides. You have a question? Another one?
What would be the example of the 2 other families besides the legumes and grasses?
Yeah, brassicas are another one. Some plants don't support a mycorrhizae network, but they're still there. They influence it, right? I personally go to an area and I look around and see what grows without anybody's help. At which point I try to find cultivated crops from those families because those families kind of do pretty good there without anybody supporting them. Connected back to tillage real fast, depending on what you guys are growing, most vegetable crops are not herbs. Mint is another family, right? Most vegetable crops are not herbs. They're early successional weeds. Lettuce is a thistle. The reason why we grow it at scale agriculture isn't because it's the healthiest for us. It's because it grows well in disturbed soil that doesn't require much except, you see what I mean? It's a whole other thing. We catch it before it goes to thorn or to seed. But as a plant in a family, thistles, right, that plant isn't the best for us. It's just what can grow there. And it's a shift of management practices. Plantains, which is another one that's easy. It's in everybody's parks. It's probably here on property. You can find it in a few minutes. Plantain is, it's also known as white man's footprint. There's a really great book called Ethnobotany of Native Americans. There's no pictures in it at all, and it is fine print. But it will tell you what each of the traditional people groups in the United States used and cultivated and how they utilize those plants for both medicine and food and utilitarian purposes.
What is the name of the book?
It's called Ethnobotany. Ethno is just like people groups, right? Botany is how they use plants. of North America, Ethnobotany of Native North America, I believe is the title.
I'm sorry.
No, for four.
I was talking to a guy at Midwest Bioag, Midwest, and then some guys at Advancing EcoAg, and
That's John Kemp's group.
Yeah.
Great podcast, by the way.
And I don't know if this is just the idea for larger scale growers that it's like, how do we make that much compost to cover a field of this size? But a lot of when you look into their products, and it's just a breakdown of some sort of sugar input, and then they add in the biology, and they will do a sugar input once every 2 weeks, type of thing, and it's
it's not worth it.
does that? Well, they think it is.
Well, that's because they're selling it to you.
Well, the idea that you could use that, or you could just buy your own sugar, technically. But
Yeah.
is
Most of it is blackstrap molasses, unsulfured blackstrap molasses,
so? But does that?
which is cheap.
Can you actually see a difference if you're
So if you already use a
Rhizo
sugar,
or something like
if
that?
you're using a sugar, it's feeding the bacteria. It's not promoting the fungal populations.
I've heard though that the fungal population then, Kemp says fungal population would then buy in on the bacteria, and that's how you push both. So you'll have a bacterial bloom, and then you'll have a fungal bloom after that because it's
They have something to work with.
right.
Right. That still requires the fungi to be there in the first place. And what they're finding now is that it doesn't matter how much sugar you throw out there, how much bacteria you throw out there. If you're waiting for a spore to fly through the air and land on your field, and then all the other factors, it's going to take way more. That's why you hear it's like
Fungal, they're not just doing one. They have their mycogenesis, and I haven't really used much of their products, but but does is that really a?
So let me.
Yeah.
I actually know John too.
Okay.
So, indigenous is always the best. Indigenous means of place. If you're growing roots, you're of place. It's how you know you're a grower and not a tourist. Okay, you're growing roots into that space, that community, that ecosystem. You as a macro as well as the micros. Most of the biology that is out there on the market is truly a recipe of someone going, okay, we're a little teaspoon of this, 15 pounds of filler. If it says 99%, 93% inert medium, that's filler, which means that bag is 1 teaspoon of biology. That's what you're paying for. There's more biology in this, and that's how I know, right, how to process this over and to make it into something more. For the last year, I've been bringing a product onto the market. The source material for that product is not from a worm. It's from a different creature. No, it's a little different. Well, this is what's become created because of it. Oh, it's right here. All right. So when I go out, do my little birding, right? This is fun. You go to the store, right, and shake it out. That's how I walk through the woods. How do you guys? Do you leave it nicer than you found it or not? Right? We can pass this around. This is just screened, but it's from a different kind of animal. That its sole purpose is what's called a mycophage. It eats fungi, packs them together in humic crystals, and leaves a stable human, Humin, just waiting for it to be into the appropriate spot. The only reason we're able to make this as a product is because an ecosystem was preserved and nurtured to be able to have this at scale. And now we can produce it at scale. That's not user-friendly for most people. This is a liquid extract of that, stabilized with other goodies, right? So that the biology is then paired and shelf-stable and can go out. Yeah, that's the cool part. That's what takes you a long time to figure this out, Tracy. But because of that, it's now applicable, user-friendly, at scale for growers. If you don't have that, nature-wise, the best is a compost pillar, worms, millipedes, the whole spectrum of the soil food web, the biology. All right, so this is actually a really great shot. This here, do you see how dense the mineral is?
Yeah.
Okay, so no matter what, we can tell that that structure, see how fine the particles are? Okay? This is what most dirt looks like, right?
Dirt, yeah.
Well, this is where, no, I agreed. This is where it's so fine and it's just mineral. That would be dirt, right? It's only because we can look closer to see if the biology is there, just in that tight mineral structure. That's why the observation tools are so important. 100%, everybody honestly needs a microscope. If you utilize it for a season, 2 seasons, you shouldn't need to by 3 because you actually have macro scale observable evidence that the microbe scales is taken care of. That's when you go out at nighttime and the worms are on the surface and then making more air and water passages. You'll see these little mounds of droppings. That's the worms telling you, we're good down here, putting it up on the surface. This here is a nurtured worm bin. You guys can see. If you peel down the sides, you'll be able to see them move around, creating those air and water passages. It's all establishing what we normally don't see because we're on the surface. Got to look below. More questions? Yeah.
Sorry. Like a lot of growers that I see, especially in the small market scale situation, I mean, it's like 50 tons of compost between every crop, and they're just layering it on.
Right.
Is that what we're talking about here? Or
Hope not.
About a smaller input and having better results, or like, because it's like, okay, I look at this and it's like, it's dead. I'm not saying that this is.
No, I understand. It's dead.
And it's like, okay, it's dead. Okay, now what? Do I call up compost, or do I do this? Do I have to? Because I've tried to use inoculants, and it didn't work. The only thing I've seen is just that recently when I layer the sugar on top with the stimulant on with the microbes, and I've seen some like that root hair starting to grow out more.
Excellent.
But then it's like, is that what we're talking about, or?
So the ideal, and I'm also going to say the greatest return on investment, is to put the biology with your seed as a seed coat or in furrow, or working it into your beds and then planting into those spots. Compost, most compost is sterilized and you don't really, it's rot. Most compost is rot. Like, it's gross, some of it. They're trying to get rid of a waste stream. If you put a ton of that out there, or 50 tons out there, you're also taking what is in that and putting it into that system. Lynn Hoag, he had a whole bed. Tracy I did a class a couple months ago. That whole bed died, and it's 100% connected to the compost he put on it. It was toxic, right? And so that's why if you create your own, you know what you're putting into it. If you're utilizing a product like this, by putting it in with the seed or on the seed and then planting, that seed will take what biology is there and say, oh, I need more of you, and nurture that population, right? And then that's what creates what's called a rhizo sheath. If you pull up your roots, and I don't mean pull up, right? You open the ground, right? You break it up. You're trying to keep everything intact, but you loosen that, and you pull up your roots. If they're naked, they have no microbiome that they're working with.
Are there certain plants that would not have that as opposed to others? For instance, like radishes.
Radishes will still develop it.
I mean, it's usually like hackberry turnips. It's just this sheeting and sheeting.
Yeah, but right at the base, you'll have the feeder root hairs and everything coming out. They will be covered with what's called a rhizo sheath. It's a shield, a protective layer to them.
Like on green beans, I don't usually see that even.
Then you're missing the biology that wants to protect your green beans. Because that's the whole job. The plant literally through those aggregates will be pulling that closer and closer to it, right? That's the fungal community bringing those resources in. At that point, the hairs are covered in, I believe I have a sample from a recent farm, And we couldn't find any in acreage of vineyards. Couldn't find it at all in the vineyard. But I could find it in the weeds, right? And it was in the weeds that were at the end of turns where they turn off the sprayer and turn around, and then they hit that sprayer again, death, for another 200 yards. But in those unkilled corners, we were able to find, Rhizo Sheaths. Anyways, I'll pull these out, but you can see some of the roots.
So this is an example of the Rhizo Sheath.
This one in particular isn't. It's just the first one on top.
You know, if it's naked, then you don't have it.
A lot of people will show pictures of their roots and they're naked. That just means everything's water-soluble nutrition. It tells you that they're a salt grower. They're using fertilizers and not fertility. This is how soil grows. And then what happens is as that plant dies, right, chop and drop, or you're not destroying that ecosystem, that biology then has the path of least resistant, and that becomes the next air and water passage. That's the path of least resistance for the worm to work its way through, eating the whole time, coming out the back end.
When you're moving across, then it sounds like what you're describing is not removing.
You're removing the surface. You're reaping the harvest, not destroying where the time is stored.
Arugula, right? Very
Okay.
big root.
Yeah.
You're gonna turn that bed over.
Okay.
Get ready for your next planting, whether it's another crop in your cycle or whether it's another successive planting of arugula. In my experience, if you don't do a fairly thorough job of removing those roots, you're going to have arugula there for the rest of your life.
Yep. Wait, real quick, because nothing's there to eat it.
Okay.
The gentleman that I was telling you about who grows indoor cannabis full, right? That's a medium-story woody plant. It's a bush.
Yeah.
Okay? Real roots, right? At that point, there's no root. Like, literally, he has a dance. This is where the next one goes. This is where the next one goes. 9-week cycle. Then he plants in the same spot the one before.
You're not removing the roots.
Nope. Not even at all. No till.
See, I have what you described about worms. I walk in one of my hoop houses at night.
Yeah.
And you can
It's
see
alive.
the floor moving.
Yeah, that's good.
That same greenhouse.
Yep.
If I don't pull out those roots of the arugula, I have arugula growing there.
What, except for worms, because worms are what's called a trophic level, right? At that point, we shift over to like springtails, pill bugs.
Lots of pill bugs.
Okay, exactly. They're the ones breaking down that other organic material. So this is where the complete ecosystem comes in. I made a pack a long time ago with gophers, right? Gophers are macro. Yeah. Gophers are macro scale. They're mammals, right? Macro scale, they create air and water passages. And then I do a lot of trench work, and when you do a trench, you can see that gopher move through right into hardpan. That's why they're there. Like, if you have soil that is aerated and healthy, they dig and the tunnels collapse on them. They don't want to be there. It's kind of boring.
I think one of the things you might be missing is that your soil's so good that plant just wants to re-tap in and the microbes will just re-hook to it, like the arugula. The only way I've seen it actually work is that if you actually tarp that bed for a week or two so that it doesn't have the sunlight so that your things can come back. But you're right. I mean, if you're going to flip it, that soil's so good it'll just replant itself.
So I can attest to the fact I'm a third-generation farmer, and I can say that the things that he's saying, the problem is, the reason why he's wrong is because...
No, good. This is why I have worked with industry.
The reason why he's wrong is because agricultural teaches you to not hear what he's saying.
Mm-hmm.
I'm just telling you that, I'm a third-generation farmer, and everything in agriculture tells you that what he's saying can't be true, and it makes it so we can't take and apply those things in our individual situations.
Total paradigm shift.
Real quick, third generation, right? This is that rule of three.
It's hard.
It's hard.
It's hard to listen to this, especially when you've been in the industry. And the things you've been taught and the things you've learned, and you're just like, it's not right. It doesn't make sense. And you just fight it, and I'm saying, I did it. I had 60 feet of soil. It was alkaline soil. We took and we had a feedlot next door. We put 40 semi-loads on a 20-acre area. And we have mint slugs, we have the stubble left over. We deep plow it, and the next spring, you look, and you couldn't find any residue anywhere.
Are you talking about arugula or
No, I'm just talking about stubble.
Biomass.
My point is that the 60 feet of soil was nutrient deficient in almost everything, and it was like a bonfire, and it just ate that stuff up, and it was gone. So the stuff, my point is not the same as about the arugula.
Okay, I was like,
no,
wait a minute.
I see what you're saying, but there was no nutrition left. There was no nutrition left in that soil. Didn't matter how much I put in, it got eaten up. It was...
And this is where the diversity also, that was the nicest way to tell everybody I'm right. I appreciate that very much. I'm like, yeah, I'm okay to be wrong too, but I think he just said I'm right, and I just want to make sure real fast. I honestly have spent a year bringing this to market and doing all the research because wherever the sample is that's moving around, right? That material, it's not worm casting.
It looks like it.
I know.
I'm not saying it is, I'm saying it looks like it.
That sample is from source. The problem is we've killed the creatures. We've destroyed the habitat. If you're waiting for this creature to wander around and then to also be at such a scale to repopulate that habitat, yeah. And then it gets run over on the road, right? This is where I then had to spend this whole last year going, well, that's the ideal. And then I heard everybody's problem of why that's not going to work. And then it had to become, well, what kind of pump? What kind of this? What kind of all the actual logistics? What kind of equipment? The how part. And that is why it's now a liquid. Because I can't... No, it's human. It really is. That's good, healthy soil. Yeah, that might have been the shaker.
Did I bring the right shaker?
And this is where a lot of the stuff in Scripture really does cross over, because this is the fat of the land. When we talk about long-chain triglycerides, right? That's what that is. Like, this is the fungal component. This is the fat of the land. This is go out and reap. This all has understanding, but it's common sense understanding because it's a shared sensory experience with the rest of life. And life, for me, the capital L, is really local, loving life, and that should include you. And if you don't have that L, it's just iffy. It's true. No, I'm teaching class. What are you doing? All right. We did go a little extra on time. I'm happy to stick around and answer some more questions. Thank you guys so much for the positive energy and the wonderful... WeGrowWith.co. Contact information. What was the last part?
I was just going to ask you the same thing you're about to say.
WeGrowWith.co. All the contact information for myself. There's also purchasing options for the different kits. Thank you very much for this. There's some educational components that reinforce some of the things that we already talked about, but it really is about understanding our role as stewards to nurture a system that we would want to live into. And land is really dead dirt, life, and land, and that dead dirt put back together.
Just a quick question, for someone who really doesn't know what he's looking at in the microscope.
Okay.
Just for a basic start, you're looking for, if I'm just looking really quick, I'm looking for life, and then I'm going to learn from there, of course. I'm never going to stop learning. But just take soil from my garden, look at it real quick.
Take it. So right now, if you're just learning, it's compare and contrast. Take a sample from your dirt driveway, right? Take a sample from your bed. It's the same minerals, right? Depending on what you've added. But at that point, you'll have a comparison of the two. At that point, it really comes down to not only seeing the diversity of size and structures, but also the diversity of the creatures that are there interacting. That's when the amoebas come, and they'll just eat that stuff up, poop out all the rest. This is that progression through the trophic levels. And then when you get to a place you don't have to use a microscope anymore because you can see it at the macro level. Like that's the beauty of it, and we can walk in a garden again. So, you're welcome.
I don't know if anybody else has a
Question? And by a little bit, like a handful. You don't have to have buckets and buckets. It's not. And at that point, you would take it, like that bag, and have an extraction, right, of that material. And then you could take it with a backpack sprayer, or depending on your equipment, and spray that in. The variables are you have to take from healthy to put it into a healthy ecosystem. If you don't know about your environment and your ecosystem, then it becomes a little sketchy. You can honestly send... There's a place on the coast that I collect sulfur from. Not everybody can do that, and so that's why I work with, other companies. But at that point, I can send it off for like $35 and know exactly what's in this, and then my little rock hammer and a little bucket, and I'm good to go. You see what I mean? So it's worth the money to pay for the knowledge so that you can use that resource whenever you need to. Yes, ma'am. Well, first of all, it's human, and then we separate it. When we separate it, you separate by acid and get fulvics, or you separate it by water and you get humics.
Okay. Now, with the acid, because I help people with autoimmune disease, and I know that the fulvic and the humic minerals are good at bringing back that cellular membrane.
Yep.
So my question is, are there better and worse things? Because now there's one man who is selling 15 million-year-old dirt for a lot of money, and I thought to myself, wow, I wonder if we should garden. Does that just kind of...
So most fulvics right now at mass scale market are made of what is called brown coal. It's soft coal called langbonite or linardite. At that point, you're dealing with a... Linder.
Start from the beginning, I'm sorry.
Lindsay, will you look that up, please? At that point, they take it, they wash it with other chemicals, and then they have it. No. What are you So, if you go right now to a log, right? That's starting to be recomposed, not recomposed, right? Repurposed. At that point, your water will turn brown. All that water soluble will be going into that water. This is where our knowledge of place, our understanding of our ecosystems, I've worked with, there was a vineyard that I worked with, and they're like, these spots are all dead. Spot here, spot here, spot here, spot here. Like huge dead zones in the vines. This guy's the new manager. What's going on? He's talking to the chemical people. Well, you need more of this and more of this. And I'm going, hey, 2 years ago they did burns. You have massive burn piles where they put all the vines in with all the metal. They burn that all up. It's full of iron oxides. It's super nuked and torched that field here and here and here and here and here. Like you're pumping chemicals on, but the whole ecosystem in those spots were crucified, right? They were under that crucible and burned up. And so at that point, it's because that person was just passing through, like an agricultural tourist, versus someone who had roots in that area that drove down that road for years and years that could say, that's why. Don't add the chemicals, put the biology back into those spaces. Yes, sir?
When we were talking earlier about tillage, and I was thinking about, was it trophics?
The trophic levels.
Our various crops will fit into that trophic level,
yep.
And that would dictate our pathway for cultivation of
yep.
that crop. For instance, you mentioned lettuces from
yep.
the thistle family.
then you're going to have a bacterially dominated soil, and it's going to do great for lettuce.
Is a pioneering sort of redemptive type plant.
And think of it from the seed perspective. This is a wind-blown seed that finds a spot, goes, hey, I'll grow here.
Because nobody else is competing with you.
Covered in thorns, puts out a ton of seeds, right? Usually a single taproot, doesn't really do much for else. Does a little mining, it adds organic material, and then it also creates a decrease of that compaction because...
For us as caretakers, if that's the type of crop we want, then we have to cooperate with that to produce that kind of environment.
Correct.
Which would be bacterial dominant.
Which happens naturally when you flip a bed and do the tillage. And that's why the boom. But at that point, you can reestablish the biology by adding it back into it and nurturing that.
But now, when you're mentioning we need to assess what's our pathway.
Correct.
We can cooperate with the system and say the pathway I want to steer this in is to grow lettuce. So therefore, I need to follow that.
Yeah. So it's called a scale of succession. And it really is about a succession of what success is. If you would like to stay at this because of what you're doing, then that's what you're promoting. If you're wanting to promote greater diversity, this is where it's a little tricky too. If you've ever heard you need a 1-to-1 ratio of fungi to bacteria, that's wrong. It actually is not even. Our agricultural land is so blank slate. It's not old growth, but undisturbed prairie land is 15-to-1 fungal to bacteria. And it's all grasses with huge 150 diversity of plants, but we call it a grassland, right? And so this idea that it needs to be this is actually because we're so used to seeing unhealthy, we don't know what it looks like when it's thriving unless you go to places where,
It's hard to find that in the United States where it hasn't been farmed and hasn't been killed because that grass
look at those.
brought so much life.
So those are diatoms. You guys ever heard of diatomaceous earth? That's what. So diatomaceous earth is these guys fossilized, just the mineral. It's high silica. So that is a living piece of liquid crystal silica that is photosynthesizing.
Which are they, green one?
Yeah. Look, and so this is also. I appreciate you guys on time right here, but you guys see, it's sweeping. This is creating aggregation. It's the wiggle that's pushing everything, and it's called Bio-Turbidity, like water, right? It's creating, right, and moving and compressing. Yeah, this is a diatom.
The yellow one?
Well, it's green, but yeah. It's okay. That's really cool. Nice find.
Yeah, it was under dried horse manure in a riparian zone.
Okay, riparian zone next to the creek over there. You find a lot of them in water. There's a ton of them in ocean water. They come in different shapes, different sizes. This is part of that diversity, but it's actually a really cool find. So nice.
Yeah.
And if it's green, it's photosynthesizing. Yeah. Mm-hmm.
Yeah, when you do grow a cover crop, you're not going to kill. What do you do about it?
Now, at that point, depending on what you're growing and what your cash crop is, if you can leave it there as a biennial or perennial, like if you're doing corn, right? If you're doing corn, you come in with the harvester higher and leave everything just as is, right? If you're going through that system and you're needing to go right on the ground, well, then at that point, you're either going to need to terminate, right, kill it, and you can do that with roller crimpers, right, that go through and they break the stalks, right? The plant now doesn't have access to the sunshine. It will biodegrade at that point and become nutrient or food cycling. At that point, you can use the same material because it's so full of fungi, and it will help break that down. I'm doing a fun trial right now. Most people don't think it's fun. I think it's fun. Dog food is mostly corn and undigestible stuff going through. I use this out on cornfields all the time. It's super great. So I started to spray backyard at my grandma's house, at which point the dog's result, right, the dog's doo, right, left out there, turns into white clouds of fungi, which means that the nutrient, which was indigestible, which is why it passed through that trophic level of the dog, comes out the other end, is now being utilized by the fungi and saprophytes to break that down and create the nutrition that's still in there and bring it into the soil. It's kind of weird to go out in the morning and see a bunch of white clouds all over the place, but I don't have to pick up dog poop.
So then you would plant no-till in that,
Yeah.
in the no-till planters.
Yep.
Then,
And I would inoculate seed when you till or drip in furrow.
with your product.
Well, yeah, any biological mine is the best because it's nature made.
Yeah, and then what about if the weeds go first?
It depends on what weed it is. The weeds, there's a great book. No, that's not the one I was thinking. I was thinking When Weeds Talk. And you can go through a company called Acres USA. They've been around for 50 years. I was just at their conference in Cincinnati. It's kind of spiral-bound copied paper, right? And it goes through and says what each one of the weeds are and what they're telling you when it comes to nutrition, what it's telling you about your land, because each one of those weeds is there for a reason. Dandelions is an example, right? Everybody can visualize a dandelion is telling you that your soil's low in calcium. Yeah, exactly. And so if you know that, then you could add not a mineral calcium, not a mineral calcium, but you can add a water soluble or the calcium that's built up into the biology. Yeah. Yep, it's all deficiencies. Nature wants to be the most thriving apex environment, full succession, old growth forest with 5,000-year-old trees, right? I mean, that's what it desires, and at that point, it's a 300 to 800 to 1 ratio between fungi and bacteria. Like, that's why the ground's squishy. You're springing off of that.
Copenhagen made a suggestion that if we took our sweepings of our personal life and added it in, that we would get a response in our biology related to our own
It's true. Now, at that point, it really comes down to what your certifying agency is. A lot of the times you can't do that, depending on... Yeah, it adds our genetic material into that, which then actually pairs that through epigenetics to what's ideal for us. It's kind of amazing. Yeah. Yeah, that's exactly what it is. And that's when you go down and pick some and eat it because your body was in tune to it.
One last kind of question, comment. Wouldn't it be oversimplifying to just say, if you make your own compost using good organic ingredients and you mulch between your rows with wood chip or something like that, you're going to get a balance of fungi, bacteria that's adequate for general farming? I mean, is that just too oversimplified? Because that's...
No, in general, that's really great. The variable is it just depends if the biology is there in the first place. And depending on what your source materials are that are going into your compost, if you're using like alfalfa as a green or material like that, if that alfalfa has been used in its growing process, had a knockdown agent like glyphosate put onto it, then that is actually into there and you're putting that into your compost. It passes through the system. And then you put that on your garden, and some people's gardens don't grow because that herbicide, that antibiotic, binds to those trace elements and will not let that plant grow. That's why it's used that way. Yeah, no. Exactly. You're welcome very much. Thank you very much. Could you say that to her? If you guys have like a review or a comment or anything like that about the class or the information, I'd love to hear it and put it online. Yeah, When we talk, it's great. It's totally a boring read. No pictures, no nothing. But if you know what it is and can look up the scientific name, it gives you so much interesting information, and it's paired to pH and biology and the nutrition, and yeah, it's pretty great.
Cover crop, if you need to plant the seeds before the cover crops are at senescence.
It's just that's your growing practice. I don't know. Depends on what you're growing.
You can't terminate it because it's not reached senescence yet.
Right. So some of the cedars can cut through a thatch layer, and by having that thatch layer on, it'll actually create a blanket which will have a higher ground temperature. So depending on where you're growing and how your growing season is, that might increase your ground temperature, allows you to plant earlier, in the season.
Go ahead and plant. Some things don't terminate.
Correct. Yeah. Yeah. You just want to make sure that your purpose of why you're growing is still achieved. You don't want it to be overwhelmed with, right? Yeah. So.